Welcome to this lecture on systems biology. Today, we are going to explore a monumental shift in how we understand the machinery of life. Instead of looking at isolated parts, we will learn how to view the cell as a massive, interconnected network. By the end of this video, you will understand how computational models can predict complex biological behaviors and why this is the ultimate foundation for the future of personalized medicine. Let us dive right in. To truly appreciate systems biology, imagine you are trying to understand how a massive, complex city works. If you only look at a single brick from one building, you would never understand the traffic patterns, the power grid, or the economy. For decades, traditional biology worked exactly like this, studying one gene or one protein at a time, in complete isolation. Systems biology changes this completely. It zooms out to look at the entire city of the cell, all at once. We focus on how all the different parts interact to create a living, breathing organism. Now that we understand the big picture, let us look at the formal definition. Systems biology is a highly interdisciplinary field that models complex interactions using a holistic approach, rather than a reductionist one. It integrates massive amounts of data from genomics, transcriptomics, and proteomics to map out intricate biochemical networks. A key focus here is identifying emergent properties. These are complex behaviors that only appear when individual components interact. For example, consciousness is an emergent property of the brain. You cannot find it by looking at a single isolated neuron. By combining biology with computer science and mathematics, we can predict how these systems behave. This holistic approach leads us to a crucial question. Why does systems biology matter in the real world? It is absolutely critical for modern medicine, especially when tackling complex diseases like cancer, Alzheimer's, or diabetes. These diseases are almost never caused by a single broken gene. Instead, they represent a massive, system-wide failure of interconnected networks. If a doctor targets just one protein, the biological network will often find a detour around the blocked pathway, making the drug useless. Systems biology allows scientists to map these detours and simulate drug reactions on a computer, paving the way for perfectly tailored, personalized medicine. Understanding why this matters brings us to how we actually study these networks. Biological systems are not static. They are highly dynamic and constantly changing over time. To understand these changes, we rely on mathematical equations called ordinary differential equations, or ODEs. One of the most fundamental models in systems biology describes how the concentration of a specific protein changes inside a living cell. We model this by looking at a delicate balance: how fast the protein is being synthesized versus how fast it is being degraded. Simply put, the net change in protein is equal to the amount being created minus the amount being destroyed. Let us translate that concept into our first mathematical model. This formula represents the rate of change of protein concentration over time. The term on the left, dP by dt, is that rate of change. On the right side, K syn represents the basal synthesis rate, which is how fast the cell actively produces new copies of the protein. The term Kdeg is the degradation rate constant, dictating how quickly the protein is naturally broken down. If the synthesis rate increases, the overall protein concentration will rise. Conversely, if the degradation rate increases, the protein is destroyed faster, causing the overall concentration to drop. Now that we have our formula, Let us see this with a concrete example. Imagine a cell producing a structural protein at a constant synthesis rate of 10 molecules per second. The degradation rate constant is 0.05 per second. We want to find the steady state concentration, which is when the amount of protein stops changing. To do this, we set the rate of change to 0. We substitute 10 for the synthesis rate and 0.05 for the degradation rate. Moving the degradation term to the other side and dividing, we find that the steady state concentration is 200 molecules. This means the opposing forces of creation and destruction perfectly balance out at exactly 200 proteins. Now that we have successfully calculated a steady state value, let us understand the general mathematical proof behind it. We start with our fundamental differential equation. Steady state occurs when the rate of production perfectly balances the rate of destruction, meaning the overall rate of change is exactly 0. By setting the left side to 0, we transform our dynamic differential equation into a simple algebraic equation. We denote the concentration as PSS to specify it is at steady state equilibrium. Moving the degradation term to the other side explicitly shows that the total rate of destruction matches the total rate of creation. Finally, dividing by the degradation rate constant gives us a universal formula for the steady state of any simple biological component. So, we have proven how a simple single-component system works. This brings us to a more realistic scenario. In reality, proteins are not made out of thin air. They are part of a two-step genetic network. First, a gene is transcribed into messenger RNA, or mRNA. Then, that mRNA is translated into a functional protein. Because the production of the protein directly depends on how much mRNA is currently present in the cell, we cannot just use one equation. We must model this as a system of two connected differential equations, linking the dynamics of mRNA and protein together. To model this two-step process, we first meet a formula for the messenger RNA. This equation represents the dynamics of the mRNA, denoted by the symbol M. Conceptually, it is very similar to our previous model. The term alpha M is the constant rate at which the DNA is being transcribed into new mRNA molecules. The term beta M represents the rate at which the mRNA is being degraded by enzymes in the cell. If the transcription rate, alpha M, increases, the pool of mRNA grows. If the degradation rate, beta M, increases, the mRNA is cleared away more rapidly. Let us apply this new formula to a specific example. We are given that the mRNA is synthesized at a rate of 5 molecules per minute, and it degrades at a rate of 0.1 per minute. We want to find the steady-state concentration of the mRNA. Just like before, we set the derivative to 0 to find the point of stability. We plug in 5 for alpha M and 0.1 for beta M. Rearranging the terms and dividing 5 by 0.1, we find that the steady-state concentration is 50 molecules. This means the cell maintains a stable pool of exactly 50 mRNA instructions at any given time. Having solved for the mRNA concentration, let us quickly verify the mathematical reasoning. This chunk of the proof establishes the general formula for mRNA steady state. We start with the dynamic equation and set the rate of change to 0. By moving the degradation term to the left side, we mathematically enforce that the rate of mRNA breakdown perfectly equals the rate of transcription. Dividing by the degradation rate beta M gives us the synthesis rate divided by the degradation rate. This proves that the same intuitive balance we saw in proteins applies directly to mRNA. We will now use this stable pool size to figure out the final protein concentration. Now that we have our stable pool of mRNA, we can introduce the formula for the second step of our network. This equation models the rate of change of the final protein concentration. Notice how the two equations are linked. The synthesis term is now alpha P multiplied by MSS. This means the rate of protein creation directly depends on the translation rate, alpha P, multiplied by the amount of mRNA available. The degradation term, beta P, represents the natural breakdown of the protein. If the cell produces more mRNA, or if the translation rate increases, the total rate of protein synthesis will scale up accordingly.
Let us complete our two-step example by calculating the final protein concentration. We're given a translation rate of 2 proteins per mRNA per minute and a protein degradation rate of 0.04 per minute. We also plug in the 50 mRNA molecules we calculated earlier. Setting the rate of change to 0, our synthesis term becomes 2 times 50, which is 100. This represents 100 new proteins created every minute from the entire mRNA pool. Dividing this total production rate of 100 by the degradation rate of 0.04 yields our final answer. The cell maintains a steady state of 2,500 protein molecules. To wrap up the mathematics of our two-step network, let us look at the final proof for the protein steady state. We start with our linked differential equation and set the derivative to 0. We move the degradation term to the other side, showing that the total breakdown of protein equals the total translation from the mRNA pool. By dividing by the protein degradation rate, beta P, we get our final algebraic formula. The intuition here is powerful. The final protein concentration is directly proportional to the mRNA concentration and the translation rate, but inversely proportional to its own degradation rate. This perfectly captures the flow of information through the biological network. So we have proven the math behind the two-step network. This brings us to a beautiful biological concept revealed by our numbers: biological amplification. In our example, the cell only maintained a small pool of 50 mRNA molecules. Yet, this resulted in a massive pool of 2,500 functional proteins. How does this happen? Because each mRNA acts as a template that can be read by the cell over and over again. Since the protein degrades relatively slowly compared to how fast it is translated, the biological system effectively amplifies a tiny initial genetic signal into a massive, robust physical response. Let us visualize everything we have just learned in this diagram. You are looking at the fundamental flow of information in a biological network. On the far left, we start with the DNA, which is transcribed into the mRNA in the center. Notice the arrow moving from the mRNA to the folded protein on the right, representing translation. Crucially, look at the downward-pointing red arrows beneath both the mRNA and the protein. These represent the degradation pathways we modeled mathematically. This diagram perfectly captures the dynamic balance of systems biology: constant creation flowing left to right, perfectly countered by constant destruction pulling downward, maintaining the delicate homeostasis of life. We have covered a lot of ground today, so let us review the key takeaways. First, systems biology represents a monumental shift from studying isolated parts to understanding massive, interconnected biological networks. Second, we learned that mathematical models, specifically ordinary differential equations, allow us to predict how these complex systems behave over time. Third, we saw how the delicate balance of synthesis and degradation creates a stable, steady state, keeping the cell alive. Finally, Remember that mastering these computational models is not just an academic exercise. It is the ultimate key to unlocking advanced, personalized treatments for complex diseases. You now have a solid foundation in systems biology.